While they're setting up, I'll speak. So I was asked to speak today about microscopy and the shared facilities at the University of Waterloo. And I've been a part of quite a few different equipment grants to improve the facilities that we have at Waterloo. In the Watt Lab, we have 2 SEMs and we have a new TEM, transmission electron microscopy and scanning electron microscopy. We have 2 confocal microscopes in the Faculty of Science, and a current interest is in a development of correlative microscopy. And correlative microscopy is where you can take a shuttle and find and take the same image under scanning electron microscopy, that incredible structural details, and then also overlay that with the same confocal image, where you can use fluorescent probes. So you know exactly where, for example, in life sciences, where fluorescent proteins are expressed and have that fine structural detail from SEM. And actually, it's pretty easy to do. There are both services, technical support in the Watt Lab for scanning electron microscopy. And in the Faculty of Science, there is also support. And there is a user fee, hourly user fees. And I know in the Faculty of Science with the confocal, there's also a yearly contract. And many faculty members pay into a yearly use, as much as they want to use. So I'm going to introduce our speaker because basically the expert in all of Ontario is Dan Stevens. Dan Stevens has been 9 years with Zeiss and he received the Meister Award for, I guess, the highest performing application specialist in North America, and got his PhD here locally at the University of Toronto. And Dan, I've asked Dan to speak on behalf of us to tell us about the utility of the correlative microscopy and other microscopy we have here at the University of Waterloo. So you have one of these things?
I do.
Okay, so I'll let you take over, Dan.
Good. Okay. Thank you, David. Thanks very much for the invite, everyone. So the intention of this discussion is to do a couple of things. One, to introduce you to the optical microscopy resources that are here already at Waterloo. We're going to brush over that. 2, to introduce you to some analytical imaging techniques. I'm saying introduce. I'm guessing from the content and from what I know that this is an introduction. So this is probably peripheral to what you're doing now. I'm guessing again. Maybe give you an idea of some things that could be complementary to the work you're doing now. Some new techniques in optical imaging, and then the correlative that David touched on. So I'm the application specialist, and I work with, there she is, Wendy Allen at the back there. If you want to buy a microscope, then you talk to Wendy. If you want to use a microscope, then you talk to me. There's a lot of gray zone in between the two there, but we work together to make sure everybody's microscope works. So the title of the talk, Innovations in Imaging, and the title of the whole session, I guess, when you look at innovations in imaging, if you were standing outside if you're thinking purely optical, and I'm sorry, everything here is optical. I know everybody is exposed to other, but if I'm thinking purely optical, there certainly is a progression in optical imaging, but it seems rather slow if you're standing outside of it, I'm sure. And the reality is that if we look at the past 3 years, we've got about 5 fundamentally new techniques in optical imaging which are giving us information that we never saw before. So we have this huge span of microscopy where we're using light and we're shining light through and at things, and we're seeing things at sort of optically limited resolutions. And then in the past year, there's been an explosion as a result of technologies on the camera side and technologies on the biology side that come together in the microscopy, and now we can do things we couldn't do before. The very first slide there, I should have mentioned, this is a platelet. So this is optical microscopy looking at a platelet, which is on the order of a couple microns, and we're seeing obviously protein aggregates that are around the membrane that are on the order of tens of nanometers. Okay, optical microscopy, and we'll talk about that as we go through here. Okay, so everything we're going to talk about is high-resolution fluorescence imaging almost exclusively. Just a brief show of hands, if you could. How many people do fluorescence imaging of some sort? Okay, so Other people are doing either non-optical techniques or, I think I'll very briefly say then that fluorescence microscopy, the intention is you're shining light on a sample that's been labeled with a fluorophore. The fluorophore absorbs that light and emits light of a different wavelength. And the reason that you're doing this is it allows you to target specific locations, usually in a cell or in a tissue. So we can take something that's antibody conjugated or in some way targeted to a portion of the tissue, and we can see it with very high specificity, okay? So the left side is just a typical light microscope, fluorescence microscope, if you will, epifluorescence. This is an optical sectioning technique, in this case called the apotome, but we're moving from typical epifluorescence to high resolution. The idea being that, hey, there's more detail here than what we can get when we just fire some light into the sample. And even more so, and we can go to, oh, missed a slide, that's okay. So what are the techniques that we can use to get things that are on a subcellular scale in terms of dynamics and quantitation of cells and tissues and whatnot? We have some resources at Waterloo we'll touch on. We'll touch on what we can do with those resources. I've already mentioned specifically FRAP, spectral imaging, and fluorescent correlative spectroscopy. And then we'll talk a little bit about structured illumination and localization microscopy. So that's 2 of those 5 techniques that I mentioned at the beginning where we've had this, I think it's fair to say, a bit of a revolution. Okay, here's a whole slew of, and in fact this is an old slide. So this is how much things have changed. I really should have new branches here and new techniques. But these are all the techniques that we can use to apply to get high resolution. When I say high resolution, this is a very relative term. So I'm talking submicron resolution with optical microscopy, or a better way to say it maybe is diffraction limited. So physics is actually limiting what we're seeing in terms of the resolution. On campus, the resources that we have, some of the best resources we have for optical microscopy and fluorescence, laser scanning microscope, and we have an LSM 510 and an LSM 700. And unless I'm mistaken, and there is a little bit of ascertainment bias here, I don't think that there's another confocal available on the campus proper, correct? Yeah, okay. Right, yes, and we'll touch on that. So David has a really nice new instrument. I'm biased again, of course, but in terms of all the instruments we offer, it's one of my favorite because it's super easy to use and gives great results. LSM 700, which is a point scanning confocal. For those who don't know, the concept of a point scanning confocal is that we are seeing only the in-focus fluorescence. So I showed an epifluorescence image, and I showed beside that a very nice clear image where all this out-of-focus light was sort of gone, and that's achieved using a confocal microscope, which has this simplified optical pathway, some source for the laser shining on our sample here. We have out of focus as well as in focus fluorescence emitted from the sample, and then a pinhole in a conjugated focal plane that blocks the out of focus fluorescence. And so our detector only sees what's in focus. That's the result. And more importantly, if we're going to talk about actually getting data out of things, this is the sort of thing we can do with the resulting image. So these are mitotracker red labeled mitochondria, and because we can image them and exclude this out of focus fluorescence and get this resolution out of them, we can now do volumetric analysis on the mitochondria. So we can take a look at this, and we can do this in live cells, and we can look at dynamics of mitochondria, sizes of mitochondria, structures and elongations and branching of mitochondria, and we can correlate that with disease states. That's a pretty powerful thing to do. Okay. Yike. 10 minutes? Analytical imaging with the confocal. So I think I'll breeze through this, but I do want to touch on it because this is something that you do have available. If we want to look at an object or a sample and we want to get information out of it other than just to look at it, we need an image. This is a gratuitous shot of my children to show how cute they are, but I try to use it to emphasize something that took me a long time to come to grips with, or I guess to realize, that is that I'm coming from the biology side, and if I'm used to looking at tissue and I'm used to evaluating things by saying, well, that's red and it's big and it's kind of beside this thing that's green and it's also, that's looking at my data as a picture. So the same way that I'm looking at this guy and saying, yeah, he's got a nice smile and he's, his tie's backwards. I don't know what he's doing there, but good. Really, we should be looking at these as images, and images are comprised of pixels, and pixels are comprised of values, so we have really data here. So an image is an array of data as opposed to a picture, which is, okay, I'm looking at this and it looks like it's, yes, it's positive or no, it's negative. And if we can move from the yes, it's positive, no, it's negative, looking at the picture to this more analytical concept of it, I think, in my experience, that we see that the quality of the imaging and the data improves dramatically. One thing that we can do, actually, I'm going to very briefly, I'm going to pretty much skip over this. Something that David's microscope can do that's very powerful, if you do have endogenous fluorescence in your sample, autofluorescence, or if you do have a complex fluorophore mixture, we can do spectral imaging with the LSM 700. So basically, not only can we get this high resolution optical imaging, but we can get spectral information out of each pixel. And that can tell us the contents of that pixel, or it can tell us the interactions dynamically in that pixel, that sort of thing. This is a Brainbow mouse. So the Brainbow mouse is a transgenic mouse that's been used to essentially fingerprint neurons so that we can say, okay, well, that has that color, and therefore it is this type of neuron doing this, presumably. Okay? I'm going to skip over this for the sake of time. I just want you to know that that's available. Okay, so we want to look at cells or anything alive now, and we want to look at the molecular content and what it's doing, and we want to quantify that. One technique that we can use to do that, and something that the point scanning confocals that you have available to you are wonderful for, is called FRAP, Fluorescence Recovery After Photo Bleaching. So in FRAP we have, I think I have a pointer here, right? Yeah. FRAP we have some cell, the ubiquitous, kite-shaped cell. We take that cell which fluoresces, we say, okay, this particular region of interest, we're going to bleach out that fluorescence. So we hit it with a high-power laser, high power meaning relative to what it's typically being hit with, and we get rid of all the fluorescence there. So the fluorophores are still there, but they're now inactive because they've been bleached out. And over time, because this is a live cell and because there's motion within the live cell and there's diffusion, we get recovery of that fluorescence. And once we can see the recovery of that fluorescence and we can measure the rate of change of the recovery of that fluorescence as such, then we can get diffusion coefficients. And we look to see, okay, is there one population or are there two populations? How many of these things are fixed in place? How many of these things are actually going to be recovered? We don't get full recovery. Why is that? So it gives us some insight into, at the subcellular and even molecular level, if we stretch it, what's going on inside the cell. Okay. The distant cousin to fluorescence recovery after photo-bleaching, FRAP, is fluorescence correlation spectroscopy. So we've already mentioned that in the pharmacy building, Mariana Foldvari has an instrument, a point scanning confocal as well. Her system is equipped specifically to do FCS, and she's published on this. FCS is a technique similar to FRAP in the sense that it's giving us molecular dynamics, but it's focused only on a single focal volume. So here's my schematic of the confocal again. There's my detector, my laser, my sample. And in this case, instead of looking at the whole sample, and in LSM, it's a laser scanning microscope we scan through, instead of doing that with FCS, we're parking the beam here in the sample. We're looking at a single focal volume, and we're allowing molecules to move in and out of that focal volume. So if we allow molecules to move in and out of the focal volume and they fluoresce when they're in that focal volume, we can see fluctuations in fluorescence, which are giving us an indication as to what molecules are moving through there and how fast they're moving. That's not going to work. That's fine. There's some little beads. They come through. There you go. Come on. There we go. And we get this intensity versus time plot of what's going on in that focal volume. In itself, not very informative, but if we take this intensity versus time plot and we do an autocorrelation, or sorry, a self-correlation with that, then we can get a measure of the diffusion time, or the speed of this molecule in that focal volume. We can take that, we can do it multiple colors at a time, so now we can look at molecule A, molecule B, or binding partner, or small molecule, and we can cross-correlate these things and we can see whether they're associated. So we have a red cell, also a green cell. Terrific, it's red and green. That doesn't mean anything because at the molecular level, these things may have nothing to do with one another. They're just floating around in the cytoplasm. Now we can see at the molecular level, okay, how much of a correlation is there between the red and the green? Are these things associated? Are they interacting? Okay. 5 minutes. 5 minutes to explain 2 super resolution techniques. This is my favorite sort of, I'm an undergraduate student using a microscope. Let's try and open your eyes cartoon. So a lot of people want to get things to be really big when they use a microscope because then they can see them better. Resolution and magnification are not necessarily the same thing. And we're limited here by work defined by this gentleman. So Ernst Abbe was a business partner of Carl Zeiss, and he's responsible for the Abbe sign equation, which tells us the limit of the optical microscope, what we can see through the microscope, okay? So we know from that we're working on the order of magnitude of the wavelength of light. That's our limit of resolution. So if I have 2 things, A and B, and they come together close, 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 there's some distance D at which I can't see them as 2 things, I can only see them as 1 thing, and that's determined by this equation, okay? These 2, I'm going to have to skip that. So, number of techniques in recent years that have been commercialized that are overcoming this limit. So they're not, physics still applies, good. We're not overcoming this, we're working around it, so to speak. The 2 that I'm going to talk about here are structured illumination microscopy and localization microscopy. So this is a conventional image on the left. Nice image, terrific, nice cell, nicely labeled. This is a structured illumination image on the right. So we can assume that we've collected this because it's a marketing image. I didn't do this, but it's a marketing image, so it's nice. We collected this at high resolution with great optics, and this is really at the limit of resolution of what we can achieve with optics. This is structured illumination, gives us about 2-fold better resolution than we had with the straight optics. How is it that we're doing this? Well, structured illumination microscopy involves overlaying a grid onto the cell or the object of interest. Okay, so this is a high resolution grid, so we're seeing some image that's a result of the superposition of the grid and the sample. If we consider that the sample, let's think back to this array of data concept. So we're not looking at a cell. What we're doing is looking at pixels, essentially, although they're in the cell, with different values on them. And if those values are so small that we can't see them, and we overlay a grid that is also very, very small, what we get as a result is a moire pattern. So this is my cartoon version of what's happening, okay? We have frequency A, we have frequency B. One of those frequencies is known. If we add them together, we get some image, and we can back calculate to what the frequency is in the sample. And that gives us a good idea as to what's going on at sub-resolution. I didn't say Fourier transform once in that whole explanation, although this crowd probably could have handled that. The biologists just shut down immediately when you say that. So this is a real result from Stéphane Girardin, University of Toronto. I love this because before we did this imaging, I mean, immediately he brought these samples in and he had, well, green and red are co-localized. They're both spots and they're together. And you bring this in and you look at it and you go, well, that's true, they're both spots and they're together, but guess what? There's one spot and the other spot's hugging it. So that's kind of neat. I mean, I don't know what the biological significance is, but I'm guessing there's probably is one, right? Okay. This is important, and you're going to see this, so in one way or another, I want to get through this, and this is the end. Localization microscopy, this goes even beyond structured illumination. So structured illumination, we're seeing 2-fold improvement in resolution. Localization microscopy, we're seeing a 10-fold improvement in resolution. So now we're in the order of nanometers. Localization so here we go, optical localization. What's happening here? What's allowing us to do this? This is the acquisition of an image using what's called localization microscopy. So this is what localization microscopy acquisition looks like. If you were to look at a cell, you would never see a cell, you wouldn't know what you were looking at whatsoever, and you would acquire for 5 to 10 minutes maybe a series of 10,000 images where you were seeing individual fluorophores turning on and off. I haven't mentioned to you how you do that. I don't have time to explain it, but fluorophores turn on and off. And because we're not looking at resolution now, we're looking at localization. So this is resolution, right? This is limited by the wavelength of light. If I look at only one of these dots, though, I'm no longer limited by Abbe's equation. Now I'm localizing this. How precisely can I localize this? And we can localize things very precisely. We can localize things down to a nanometer. So as long as I can localize these and then sort of sum them up, if I was to sum up all of the individual events as they appeared, I would get this. But if I mathematically represent them as points, and I represent those points based on the degree of certainty to which I can localize them, then I get the image at the bottom. Okay? Okay, 2 slides. This is exciting because now we have a bridge that we've formed between the very low ends here, so electron microscopy, TEM, SEM, where we're working on sub-nanometer resolutions or nanometer resolutions, and the optical microscopy where we used to be working on micron resolutions. So previously, hey, I can see that, that's wonderful, and I can identify that chromosome, and I can see it very clearly at the limit of light, and I can go to electron microscopy, and yeah, it's really hard to find, but I can see it at really high resolution. Is it the same thing? I don't know. How do we bring these two things together? Well, now we're talking about techniques that can bring us down to the level of electron microscopy. And not only that, but we have the mechanics, if you will. It's really just a matter of how do we physically get these things together and know where the coordinates are. We have the mechanics so that we can do this in a very reasonable, practical way. So I can go from the super resolution system, I can get this, green thing hugging a red thing. Wonderful, that's great, but I'm only seeing that one protein complex. I want to see the cellular milieu around that and see what's going on there. And then I can do this on the EM. So examples of that, platelets again, because they're very small. So great, we have fluorescence, very specific. We know what's going on. We know our protein of interest. And then here's what's going on with the EM. We can overlay those. And my PC is telling me I'm done. Yes, okay, I'm finished. Perfectly timed. Okay, so I think the last slide said this is me, and if you want to get me, I have a Twitter account at @theperfectPSF. PSF is a point spread function, which is a mathematical representation of the diffraction of light. And we have wonderful support hotline, and that lady right up there if you want to talk to her. We're around for the next, well, Wendy's around for the rest of the day, and I'm around for the next hour or so. So love to talk to you if you'd like. Okay?
Great.
Thanks, folks.
Are there any questions for Dan? Yes, Melanie.
Dan, do we currently have the ability to do structural illumination?
No, you don't. You don't have that instrument. And it's not something that people are home building. So the localization microscopy is a rather commonly home built, and given the, what Waterloo does, I would think that there's maybe somebody in a closet somewhere who's already doing this. I don't know. But SIM, I don't think you have access to on campus. I just, maybe a note there, in case there's some confusion. Structured illumination is a broad term, sort of this is tissue, this is Kleenex term for a number of things. So super resolution SIM, more specifically, you don't have access to. There's other structured illumination that gives you typical confocal imaging.
Okay. So Melanie, would that be something that would be useful for your research?
Possibly. I mean, you have to look at yeah, but then it requires very little vibration and all the rest of it. You have to do it on a quite microscope.
It requires the same anti-vibration that you use for a typical optical scope, but a little more concern about the people coming and going and the air conditioning and whatnot. But it's not sort of, you can put it on an anti-vibe float table and you're good. Yeah. That's the worst of your considerations after the money, yeah.
Okay, Zoya.
The localization microscopy doesn't require any specific fluorescence label?
Yes, thank you for asking that question, because I didn't get to say that. So I guess at first blush, from what I told you, you have a technique that does 10 nanometers and you have a technique that does several hundred nanometers. Why would you ever do the several hundred nanometers? Why not do the 10 nanometers? So that flashy that I showed on the localization microscopy, your sample has to perform the flashy. And there's a couple different ways to do that. You can do it with photoactivatable fluorophores. You can do it chemically with something called storm, where you essentially have an appropriate redox environment so that you're forcing fluorophores into a dark state primarily, and they just kind of drop out of that randomly, and you can balance that out so that you get individual fluorophores. But whatever it is, it requires that you have most of your sample dark and then occasionally get a fluorophore popping into the light state. And that can be, depending on your sample, tricky to do. So it's a practical limit at the moment, which is being, I have to say, overcome at a rather rapid pace. Yep, absolutely. You can. That's the intention. Yes. Did I answer your question?
Yeah. How specific? Not just general, but how specific?
Oh, how specific? Sorry, I didn't hear the how. So if it's an antibody label, then it's as specific as you can make your antibody. And there's a range of qualities of antibodies that you can get commercially or make yourself. You know, a good antibody, you are looking at not just the protein, but the epitope on the protein, and it's only binding to that, and that's it.
So in that sense, it's not viable for living cells.
You can do living cells as well. In that case, typically you would tag the protein of interest so that it would be expressed endogenously. So photoactivatable GFP or photoswitchable GFP, these can be used as well. Yeah. That's, POM is the technique for photoswitchable. And I talked about this as POM. STORM is the technique for the antibody labeling. There's about 20 different variations of it, but most people are calling it STORM, stochastic optical reconstruction microscopy. Okay.
David, I was wondering, could you give a couple of comments just on where the facility is located and how does somebody get in touch if they want to use something?
So in the Department of Biology, we have, it was actually purchased about year 2000, the first confocal microscope on campus. And it's still working, but it's sort of no longer going to be under service contract because it uses older technologies. Now we have solid state lasers on the newer microscope, the Personal 700. So there is a technical support person, Mishi Gro, who's in charge in the Faculty of Science for support, and all graduate students, even undergraduates, can get support in how to use the microscope. It's the same software that is used on the 510 is also used on the 700, and so it's really useful. So we upgraded the software on the 510 and the 700. And as far as I can tell, that software is actually also compatible with the Merlin Gemini 2 scanning electron microscopes too. So it's pretty amazing because you can go from, you know, using the same basic software in all three. So it seems to me that the limit to microscopy is people don't know what they need, but they think it's pretty cool because they see beautiful images. They don't know how to apply it to and these microscopes are very, very expensive. And so one of the important features, I think, is the usability of the system. And so the software that we have in these three microscopes, sets of microscopes, are compatible. So that's really useful. And they have sort of press one button and it works kind of stuff, as well as being able to do your own, like working with a camera, right? You can take the one-shot kind of deal, and then you can go in and change all the parameters if you want. So that's a really good thing. I know there are a lot of users across campus in both life science and material sciences with overlapping use requirements. And so there are a lot of microscopes that would be very useful for people to use. One of which, for example, is even the level of a stereo microscope. So I know Zeiss, but there's other vendors too, like a zoom stereo microscope, where people can look at things like live embryos, that sort of level. But you can do optical sectioning like with Ableton. Isn't that right, Dan? There's optical sectioning. So it's like, basically it's like a confocal microscope, the level of a stereo zoom microscope, where in fact you don't switch between different lenses. In fact, it's a zoom microscope. And there's enough of a working distance that you can look at living cells and living animals, right, at that resolution, with multiple fluorophores, right? So, and there's lots of different platforms that you can use. There is a lot of a package that you can use, for example, for live imaging of living cells. So some people want to work with in material science, so it doesn't really matter if there's an incubation chamber, but you know, we could in fact outfit some of these microscopes to live cell imaging using water objectives as opposed to oil, for example. And that would be very useful for some people to use, right? So, yes?
Can I actually just add a little bit? Sorry that I forgot to say, probably important to some people, the LSM 510 can be used very nicely to image surfaces or textures on materials. It doesn't have to be fluorescence. So you can get diffraction limited resolution, and even if you have sort of semi-transparent layer thickness and this sort of stuff off of your samples, I don't think it's being used for that at all, but given the amount of material stuff that goes on, I'm sure somebody out there who might be interested in it.
Yeah.
I don't know if Mishi's ever done that, so I might
Yeah.
have to have a chat. But if you're interested, then that's something that the 510 can do.
Yeah.
And some of the best images I've ever seen come out of sort of mixed. So if you have a biological ANA, you're doing biofilms or something like this on materials, that can look gorgeous on the Confocal.
Yeah. Yes.
Sorry.
So the two confocals are actually quite different. So the Zeiss 700 is actually just a actually a pretty more straightforward unit, where the optics are more uniform as far as I can tell, and that you get brighter images compared to the 510. But the 510 has meta functions for spectral analysis, which is not available on the 700. But if you go to the technical resource people, both, so in terms of I mentioned to you what's in the Faculty of Science, in the Watt Lab, under Tong Liang, he's got also a price list and technical support for doing doing more of the high resolution SEM, and there's a brand new TEM also available, and it's in the basement of the QNC building. So we have transmission electron microscope, brand new. It's been up for a few months in the QNC in the basement. We have a TEM in Department of Biology. It's an older model, and so probably I recommend people to probably use that one rather than the one that we have in biology, although it's a secondary resource. We have the two confocals in biology. One's in B1 and one's in B2, but if you contact Mishi Groh or myself, and in fact, what we want to do is set up a website where this is available so you can see what is available, the costs associated with it, the hourly costs, and where you can contact for support.
David promised me to compile all this information, and we'll put it on the CB website so people can access it and try to find out what's where and
Absolutely.
how to get there, right?
And we also, and basically because these microscopes are so expensive, right? And it's hundreds of thousands of dollars, at least $100,000 for a basic setup for some of these microscopes, to millions of dollars, multi-millions of dollars for the SEM and TEM. We need a lot of users, and we need to have a database of people who want microscopy and what your needs are, so that we can go together on collaborative grants, whether this is CFI or little bits of grants through RTI. And there's lots of opportunities there, especially if we get a consensus for what people need. And so on the website, I would, through the website or through some sort of questionnaire, get a sense of what people's needs are so that we can expand on the resources we have.
Perfect. Yeah. So I'd like to encourage people to do a bit of thinking about what are you maybe missing that would help your research, or what are you leaving Waterloo to get? Okay, that's the other kind of way of looking at it.
Yeah.
And maybe come back to us with some feedback about what would be most helpful, so we can kind of progress in this area.
So the biomedical imaging research has suffered a lot in the past decades, mainly because no access to large enough data sets.
Mm-hmm.
That's a huge problem that many institutions are trying to address. research community is trying to share images.
Mhm.
So is it that individual researchers just come and take their images, pay for it, and they go, or you're also maintaining an archive of sample images that can
I
be
think
publicly shared?
an image library would also be.
Yes.
Too.
Yeah.
Do we have
So do we have that?
I don't think there's anything formally set up, but
Okay, so that's something maybe we should
Yeah.
be doing.
yeah,
Absolutely.
that would be.
Yeah.
Yes?
Yeah, same thing here. I've actually not people who work on this,
Yeah.
Yeah.
so I feel like this would be very valuable.
I know that a lot of the, like for example, the Ontario Brain Institute is very interested in making sure that there are archives of all the material that they, through the grants that they receive, and that all that's public access.
The problem with the metadata is that the sheer amount of data is not enough for research. We need label images
Right.
to do something meaningful. So some sort of expert have to process it, put some annotation, some marking, some label.
Yeah.
That this is this, such that we can verify.
Yeah.
And so that's the main thing, I guess, one of the factors that people have not been able to
Yeah.
do.
Okay.
I-
But if we were able to establish something like that, would that be something that would be of use to researchers outside of Waterloo? Like it would be a
Yeah. I think it's
resource that we
important.
could share or.
I think what we need to is just to have a common point of contact
Yeah.
for all microscopy resources, and that we feed in and it services everyone's needs. Like other universities have like a centralized microscopy resource. Now it doesn't matter if the microscopes are all over the place, as long as that we're sort of all in communication. And I mean, we have major faculties that all want microscopy, whether this is AHS, or it's Faculty of Science or Engineering or our arts, all wanting to use different levels of microscopy. And so it would be great if there was something set up, which then could enable something like that, where we combine our efforts.
Yeah, we're going to be a lot more successful in our proposals, right? If we all team together, saying that we want something.
Yeah, I know about this because I've got like 28 people on my current grant from 11 different departments, and I can tell you it is an inordinate amount of work to make all those contacts, and also people aren't aware of what the thing is you're trying to get, right? And where it could be useful for people. So it's better that there is more information available for faculty. There's more resources for them to get help when they want to apply microscopy.
Yeah, and that's exactly something that here as a center we hope to be doing for you. Okay, so that's our role. That's something we should be doing. Okay, so come talk to me if you've got things that you're interested in. If you want to build bigger teams on things, that's one of our jobs, is to try to understand at least a little bit about what all our members are doing. We can kind of try and facilitate things like that for you.
So people here in favor of a website and a centralized resource for microscopy?
Oh,
Yeah?
absolutely, absolutely.
Okay.
Yeah.
So that's kind of like the first step,
Yeah.
is to have a website